Hello, welcome to Biot Anime. Opsonization. The term opsonization refers to the capacity of antibodies and complement components to coat dangerous antigens, that can then be recognized by antibodies or complement receptors on phagocytic cells. This mechanism will identify invading particles or antigens, by the use of specific components called opsonins. The opsonins act as markers or tags that allow recognition by the immune system of the body. An opsonin is any molecule that enhances phagocytosis by marking an antigen for an immune response or marking dead cells for recycling. Mechanism of opsonization. Opsonization of pathogens can occur via antibodies or the complement system. Antibody-mediated opsonization. Antibody-mediated opsonization by antibodies involves the coating of pathogens with antibodies, so that they are recognized and phagocytosed by innate immune cells. The complement system is only fixed by certain immunoglobulin. They are immunoglobulin M, immunoglobulin G1, 2 and 3. The site of complement binding is located on the Fc piece of the immunoglobulin molecule. CH2 domain on immunoglobulin G and CH4 domain on immunoglobulin M. Immunoglobulin combines by its two antigen binding pieces, FAB, with antigenic determinants on the surface of the microorganisms. Upon combination with antigen, the immunoglobulin G molecule undergoes specific conformational and configurational changes in the FAB2 hinge region. Phagocytic cells of all types have receptors for the immunoglobulin G molecule on their plasma membranes. The phagocytic cells' receptors for immunoglobulin G bind only to the Fc portion of the molecule, and are therefore known as Fc receptors. In case of antibody-mediated opsonization, binding of our antigen-antibody complexes to an Fc receptor on phagocytes will induce internalization of the complex and internal digestion of the pathogen in lysosomes. Generally, multiple antibodies bind to various sites on antigen, increasing the chance and efficiency in which the pathogen is engulfed in the phagosome and destroyed by lysosomes. Complement-mediated opsonization. The complement system is composed of over 30 proteins that improve the ability of antibodies and phagocytic cells to fight invading organisms. It initiates phagocytosis by opsonizing antigens. This system is also responsible for enhancing inflammation and cytolysis. Different complement pathways are 1. Classical pathway. 2. Alternative pathway. 3. Lectin pathway. Classical complement pathway. C1QRS protein will bind to antigen-antibody complex through C1q with Fc piece of bound antibody, in the presence of calcium ions, leads to sequential activation of C1R and C1s. The C1s is an esterase that can cleave C4. C4 is split into C4a, an anaphylotoxin, and C4b, which binds to cell membrane along with C1. C4b in the presence of magnesium ions will cleave C2 into C2a and C2b. C2a remains linked to cell-bound C4b and C2b, which is released into the fluid phase. C4b2a has enzymatic activity and is referred to as C3 convertase. C4 convertase splits C3 into two fragments. C3a, an anaphylotoxin, and C4b, which remains cell-bound along with C4b2 or 3b, which has enzymatic activity and is called C5 convertase. This C5 convertase will split C5 into C5a and C5b. C5b will continue with the cascade C6 and C7, then join together. A heat-stable trimolecular complex C567 is formed, part of which binds to the cell membranes and prepare it for lysis by C8 and C9, which join the reaction subsequently. Alternative complement pathway. Alternative complement pathway begins with the binding of C3b to an activator, which include bacterial endotoxin, immunoglobulin A and D, cobra venom factor, and the nephritic factor. The C3b protein is continuously generated as free state in small quantities in circulation. It is rapidly inactivated by serum protein factors H and I, but the bound C4b is protected from such inactivation. Bound C4b will then interact with factor B or C3 proactivator to form a magnesium-dependent complex called C3bb. This complex, C3bb, is cleaved by another serum protein, factor D or C3 proactivator convertase into BA and BB. The fragment BA is released into the medium. Fragment BB remains bound to C3b, forming the esterase C3BBB complex or C3 convertase. The C3 is then cleaved into C3A and C3B, and the cascade continues. Lectin complement pathway, also known as mannose binding pathway. Lectins are proteins that recognize and bind to specific carbohydrate targets. The lectin complement pathway is activated by the binding of mannose-binding lectin to mannose, the residues present on the surface of microorganisms like certain bacteria. Mannose-binding lectin protein or MBL protein is an acute phase protein produced in inflammatory responses. Its function is similar to that of C1q. After MBL binds to the foreign surface of pathogen, specific protease, MASP1 and MASP2 bind to it and form the C1-like active complex. Later it causes cleavage and activation of C4 and C2 to produce C5 convertase without the need of specific antibody binding. A number of circulating proteins like pentraxins, collectins, and ficolins also serves as opsonins. These proteins are the PRR or pattern-recognizing receptors capable of coating the microbes as an opsonin, which enhances the activity of neutrophils by several mechanisms. Example: ficolins typically recognize NA-style glucosamine residues in complex-type carbohydrates in addition to other ligands on different antigens. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon.